Thank you, and thank you for the opportunity to present my work here at this wonderful conference. So I'd like to start with a key feature of PMP22 that is already been mentioned by Dr. Kleupa, and this is the fact that PMP22 is a dosage-sensitive gene, meaning that slight alteration of its expression level can lead to severe neuropathies. Especially, we all know that duplication of the gene lead to the Charcot-Marie-Tooth, while deletion of the gene lead to an underexpression of the protein and lead to the HNPP disorder. So we really need to find a way to try to decrease the level of PMP22 protein to a physiological condition. And we implemented an approach that is based on base editor. These are new generation CRISPR-Cas editing tool. They are composed of CRISPR-Cas9 that basically scan the whole genome in the search for a protospacer sequence. And this is guided by a guide RNA. And this Cas9 is fused to deaminase enzymes that can either install a transition modification of a C to a T, in the case of cytosine base editor, or an A to a G in the case of adenine base editor. We choose these kind of genome editing tools because they have key features such as they are really precise, they are safe compared to knockout, and they are really efficient compared to other genome editing tools such as Prime Editor. And indeed, thanks to these key features, we have clinical trials based on the base editor, for example, for treating hypercholesterolemia and for treating antitrypsin deficiency. And this is how our approach is meant to work. So the PMP22 gene is transcribed normally, and the mRNA is recognized by the pre-initiation complex that start the scanning of the mRNA looking for ATG codon. The fact that actually this pre-initiation complex stop at a specific ATG is based on the context around the ATG codon. And this context is called the Cossack sequence, and there are genes with strong Cossack sequences, meaning that they are able to engage really well the pre-initiation complex and start an effective translation, while there are genes with weaker Cossack sequences that display lower translational efficiency. PMP22 has quite a strong Cossack sequence, and we aim to install single point modification via base editors and decrease its translational strengths, and thus decreasing the translation moderately of the PMP22 gene. So how we measure the translation? So we employ a reporter system that comprises a green fluorescent protein that is under the control of the PMP22 Cossack, either wild type or variants. And we also use a red fluorescent protein that basically acts as a normalizer for the signal. This whole reporter is cloned into a plasmid and transfected into HEC293 T cells. And after 2 days, we measure the Cossack strengths as ratio between EGFP and mCherry via high-content image screenings. We look for many variants for PMP22, and we found 3 that decrease the translational efficiency of about 5, 10, and 20%, and those are variant 2, 3, and 4. And these variants, as you can see, variant 2 and 4 can be installed by canonical cytosine base editor, while variant 3 can be installed by adenine base editor. Variant 3 and 4 are in the coding sequence of PMP22, while variant 2 is in the 5' UTR. Okay, next we wanted to check whether actually these installing this mutation were actually we want to validate our findings. So basically we generated a cellular model by transducing again HEC293 T cells with a lentiviral vector encoding for the wild-type PMP22 under a strong promoter, quite strong promoter, which is the EAF1 alpha promoter. And here I'm just measuring the mRNA level that obviously are really high for HEC293 transduced in this way compared to other cellular models. And next, we move with the transfecting HEC293 T cells with a base editor and a guide that allows the installation of these variants, guide 2 for variant 2, guide 3 for variant 3, and so on. So as you can see, guide 2 were able to quite efficiently store the wanted mutation, but it shows some bystander edits that are unwanted editing that occurs in the editing window of the cytosine base editor. And these are in the ATG codon, which we would like to avoid. For guide 3, again, we have some bystander edits on in the methionine, while for variant 4, we have some bystander edits. However, the last one didn't change actually the nucleotide sequence. And once we install these variants in the genomes, we show that the PMP22 protein level decreased, and in the edited sample compared to the controls, while we are not really affecting the transcription of the gene. And this is like to showing that we are affecting just the translation process while we are keeping intact the transcription level of the gene. And next, we move again into patient-derived fibroblast. We use a lentiviral vector to encoding for the base editor and the guide to install this modification. Here the efficiency were even higher. However, for the guide 3, we observe a really high bystander edits that need to be taken into consideration. And however, when we tried many different techniques, such as Western blot, also the ELISA, but all they were not successful in detecting an amount of protein because basically they express too few PMP22. So this is my recap slide. So our goal is to try to reduce the PMP22 protein level to a physiological level. We install single point modification into the COSAC sequence, aiming at decreasing the translational strengths of the COSAC of PMP22. And thus, we are trying to reach this physiological level of the protein. We identify 3 variants that decrease the translational efficiency of different amount. And we install those variants into HEC293T cells overexpressing PMP22. And actually, we see what we wanted. So meaning that we see a decrease in protein levels, and despite the mRNA levels, they remain unaffected. And finally, we have a critical requirement for the patient-derived fibroblast that is actually be able to detect really precisely the amount of protein produced by those fibroblasts. We also would like to try our approach in more relevant cellular models such as IPS, and also identify an approach that could deliver our the base editor and the guide in vivo, and validate our findings also in mouse or rat animal model. So with that, I would like to thank the CMT1 team and the whole member of the Laboratory of Translational Genomics, and also the Associazione Italiana Charcot-Marie-Tooth for sponsoring this research. Thank you.
So thank you very much. Are there questions? I don't see questions written here. You know that there is a possibility to write a question. Anyway, if something want to do it, it's possible. Same question as before. So it's a fine-tuning of protein. Did you look at the other proteins like P0 expression?
Yeah, we have a whole bunch of done a whole bunch of research on COSAC sequences. We are looking at variants of COSAC that are able both to decrease the translation but also to increase the translation, and this is key, for example, for haploinsufficient disorder, where we have the deletion or aberration of one allele and the other cannot sustain enough protein production. So we have a whole bunch of done a whole bunch of reason, and yeah, we found many actually variants that are able to either increase or repress the translation compared to the wild type. Yeah.
So CMT1A patients have 3 copies of PMP22. That means 3 COSAC sequences. And if you look at HNPP, where you only have 1 copy of the PMP22, you will have only 1 COSAC. So would it be possible that COSAC sequences can differ between the different PMP22 copies in CMT1A and also in HNPP, where there is only 1 versus control?
Yeah, the COSAC sequence of PMP22 is quite well conserved. Obviously, we will need to screen for the sequence of the patient that will possibly receive the therapy. And we can quite easily measure their translational efficiency and also tailor basically the therapy for specific for the patient. For the case of PMP22, for the case of Charcot-Marie-Tooth, we have also different variants that decrease the translation by different levels. So again, we can quite well tailor a possible therapy for the need of the patient.
Oh, there is a question here. I repeat it. Is there an opportunity to also use this method for HNPP if it works on CMT1A? I saw that you mentioned it in the first slide. Laura Offenberg.
Thank you for the question. It's actually really on point. The fact that we screen a lot of variant, or COSAC variant, and so far we haven't found one that actually is able to increase the translational efficiency compared to the wild type. We actually just check the 4 nucleotide before the ATG and 4 nucleotide after the ATG. There are many evidence that actually nucleotide in position -5 and -6 are can be really important for the translation, but so far no. Unfortunately for PMP22 we haven't any variant for increasing the translation.
Oh, there's another question. Sorry, Andrea.
the sequence. What then, if we move to humans, what do you think will be the efficiency? Like, you're going to target them all, or you think 1%, or I think we still don't know, right? It depends on depending on the tissue.
Yeah, our main focus now is to find a delivery system that is able to bring this base editor to the correct cell, basically each one cells. For now, I mean, Cleopa is doing a lot of work, but many other researchers are really trying to achieve this, the highest delivery efficiency. I would say that with, like, for example, AAV, you could reach quite good editing efficiency, and also like at 30% or 40% editing efficiency might have a phenotype. But yeah, the project is still at an early stage, so we would like to bring this to challenge this aspect in the near future. Thank you.
Thank you.